everyone, today we'll be talking about positive single-stranded RNA viruses. Positive single-stranded RNA viruses belong to the Baltimore class 4, and their genomes are very similar to cellular mRNA, which means that it can be used directly to translate proteins. The genomes of class 4 viruses have a coding region flanked by 5' and 3' coding regions. Some examples are shown below. These include Coronaviridae, the Coronaviridae family, which includes the coronavirus, the Flaviviridae family, which includes yellow fever and hepatitis C, Picornaviridae, Togaviridae, and Caliciviridae. We will be discussing how they control translation and transcription shortly. So if you look at their structures, you can see many similarities. First of all of them have a coding region shown in purple. All of them have the 5' and 3' untranslated regions shown in yellow. And all of them, not all of them actually, have the poly A tail. So Flaviviridae does not have a poly A tail, which would be shown in green otherwise. And some of them have a 5' cap shown in pink, while others do not, such as Picornaviridae and Caliciviridae. Instead, they have what we call a viral protein linked genome that acts as a primer, similar to the 5' cap. VPG. Now, what is special about positive single-stranded RNA viruses is that they never carry polymerases in their capsid. Apart from retroviruses, which are also single-stranded, instead, they use the mRNA to make an RNA-dependent RNA polymerase, which helps to synthesize the necessary RNAs. Now we're going to talk about how positive single-stranded RNA viruses control transcription. Okay, so transcription occurs in several stages. First, the positive single-stranded RNA can be translated to make the RDRP from the 5' direction to the 3' direction. And the RDRP would be synthesized either directly or as a polyprotein. With the RDRP, this is used to synthesize a negative RNA strand complementary to the positive RNA strand from the 3' to the 5' direction, at the end of which you would achieve double-stranded RNA, which is highly immunogenic. Using this negative RNA strand, positive strands can be transcribed from the 5' to 3' direction. And this happens by RDRP binding to the 3' end of binding to the 3' ends and starting synthesis in that manner, after which it is translated again into the necessary proteins. Now, there is an obvious problem, which is that translation occurs in the 5' to 3' direction, this way, while replication occurs in the 3' to 5' direction, this way. So when you get this happening, you would eventually get a collision. So the virus must adapt and evolve around this to ensure correct steps occur. Now, the poliovirus, which belongs to Picornaviridae family, avoids this by circularizing the DNA. And by circularizing the RNA, this can prevent ribosome binding and clashing. How does it do this? Well, it has several proteins which can be shown here, and essentially it uses its viral protein 3CD and host protein PCBP2 to bind to the 5' end. Then the PABP is bound to the 3' end. If this happens, you get PABP then binding to the 5' end of the PCBP2, and this joins the ends together, forming circularized DNA. And this binding also facilitates the cleavage of 3CD Pro polymerase, and when this happens, you get the detachment of 3D, and then you have the formation of RDRP for replication. And it uses the other ends as a primer, because we always know that replication is in the 3' to 5' direction. Now, we have flavivirus. So some examples of flaviviruses include hepatitis C, and again it uses a similar mechanism in which it promotes cyclization at the 5' and 3' ends to prevent ribosome binding and ensure replication takes place from the 3' to 5' direction. It is important to note that this is recognized by the replication complex RC, which includes NS5 at the 3' end to copy it. So while poliovirus uses 3D pol as RDRP, flavivirus uses NS5. Now we have the Togaviridae family. Some examples of viruses in the Togaviridae family include the Ross River virus and the chikungunya virus. The way it avoids collision is that it has a hidden promoter site hidden within the genome, and the subgenomic RNAs are not packaged within virions, and they're only produced during active replication. The difference between now let's talk about norovirus. Norovirus can also coordinate when certain proteins are made. They can produce lots of nucleic acid via replicates proteins. Both Togaviridae and noroviruses produce subgenomic mRNAs from the 3' ends, but flaviviruses and polioviruses do not produce subgenomic mRNAs from the 3' end. Okay, now on to coronaviridae. So in coronaviridae, you have a coronavirus RNA replicase which skips transcription. And by doing this, it produces a nested set of discontinuous subgenomic RNAs. In terms of how transcription can be an issue because, as we said, double-stranded RNA is highly immunogenic. So it can induce powerful interferon responses and cytokine responses, which then cause antiviral activities. So what the viruses do to avoid this detection is by having proliferation of small vesicles from the intracellular membranes or the endoplasmic reticulums for polioviruses and flaviviruses. So, and they use RNA replication in here so that it is hidden from this interferon system. Now that we have the transcribed subgenomic RNAs, we have we can undergo a translation. But it is important to control translation and ensure that more viral proteins than host proteins are produced. So how does it do this? Let's talk about three different viruses in detail: hepatitis C, poliovirus, and norovirus. Okay, so herpesvirus or hepatitis C does not have a cap structure. Which is interesting. Or VPG-like protein. It has an iris, which is known as an internal ribosome entry site at the 5' end. And it also has highly structured 5' and 3' untranslated regions for efficient translation. It does not destroy the cellular protein translation machinery. The cellular pathology is negligible, and it does not directly kill infected hepatocytes. And in translation, for both herpesvirus and poliovirus, they encode for one long open reading frame that can produce a large polyprotein, which is then cleaved into mature viral proteins via viral and host encoded proteases. So herpesvirus uses both host signal peptidase and viral serine protease. And as shown here, that's its genome. The parts marked in yellow are where cleavage by viral serine protease occurs, and the parts shown in purple are where cleavage by host signal peptidase occurs. Onto poliovirus, we see a very similar pattern. Its genome gets further cleaved. It actually has several steps of cleavage. So in the first one, you get something like this. Shown in purple is cleavage by 3C protease. Shown in yellow is cleavage by 2A protease. And eventually that's the first step, second step, third step. It also has an internal ribosome entry site, which is basically an RNA structure which allows transcriptional factors to bind. It is cap dependent. There's no methyltransferase needed. Oh, I think I meant translation factors. Translation, not transcription. I made a mistake. So poliovirus actually evades, it causes less host product protein production and thus harms the cell. So how does this stop normal cellular translational machinery from occurring? So over here we have the normal translational machinery that must be present for infection, for proteins to be produced. But what the poliovirus does is that you can use one of two steps. You can either use its 2A protease, which we previously discussed, to cleave this EIF4G component of the translational machinery, so it no longer works, and host cells cannot initiate protein translation from the capped cellular mRNAs, or you can dephosphorylate this EIF4E element. You can dephosphorylate this 4E-BP1 element, allowing it to bind to EIF4E and stopping it from binding from EIF4G. And this whole separation stops the whole translational machinery from operating. This is also a strategy used by the encephalomyocarditis virus. Now, another strategy used by picornaviridae, not poliovirus, but influenza and adenovirus, is that they just simply dephosphorylate this EIF4E element, so you cannot, so it dissociates from the from EIF4G, and it also dissociates from the M7G cap, or the guanosine cap of cellular mRNA. So the end product of this is that you have a decreased rate of protein synthesis for polio-infected cells. And if you have decreased proteins that play a major role, if you have decreased proteins, then a major role of this, which is to protect the host, is lost and host cells will die. Okay, now on to the next translational control mechanism, so norovirus. So norovirus controls translation by using VPG, or the viral protein linked genome, as a cap substitute, and it just recruits many factors which then enable it to replicate in a timely manner. Okay, so this is all of the positive single-stranded RNA viruses. I hope you enjoyed learning about how they control translation and transcription. Thank you.